Here is a zoom in picture, right? Looking at the renal corpuscle. You have again the glomerulus formed by the afferent arteriole, and then the blood flowing out from the to the efferent through the efferent arteriole, right? And then the formation of the filtrate in the glomerular capsule. Okay, but here in this particular diagram, I want you to pay attention to this structure here. So this tube here is part of the DCT. Remember, we talked about the DCT is actually physically right next to the renal corpuscle, right? So there's a reason for that. It's not only physically right next to it, it's actually the touches the afferent arteriole. So you have a special structure. See the cells right here. It looks slightly different than the rest of the cells from the DCT wall, right? So they are special type of specialized cells called macular densa cells of DCT, and they touches the backside of those cells, touches touches the specialized cells of the afferent arteriole called granular cells. So together, those kind of cells on their backside touches each other, and they form this apparatus called juxtaglomerular apparatus. So juxta meaning right next to, right? Right next to what? Right next to the glomerula. So that's why it's called juxtaglomerular apparatus or JG. So why you have those kind of cells right next to each other, why it is important. Okay, so the muscular densa cells here in the DCT can detect the changes of the sodium chloride in the DCT, right? So that's because what's in the DCT are essentially close to, already the urine form, because the filtrate is going to come out of here, is going to be modified by all those tubules and afferent loop, right? And the DCT, and so that's going to be, things going to be reabsorbed from the filtrate, and some of the ions or the molecules going to be secreted into the filtrate and to form urine. So here in the DCT, by the time it flows to this part of the DCT, it's pretty close to be the count right here. So this is pretty close to the end of the DCT. That's close to the concentration of the urine, right? The fluid inside. And so any changes in the sodium chloride, so that's electrolyte concentration of the fluid in DCT, is going to be an indication of how much, right, the electrolyte is going to be lost or, going to be in the urine. So if they say abnormal high or low level of sodium chloride based on the body's need, and they're going to signal to the granular cell right next to it via paracrine. Remember paracrine? So that's basically you secrete something and it's going to act on the cells right next to you. So that's why those two kinds of cells need to be right next to each other. And what is a granular cell going to do responding to the stimulation from macular denser cell? Well, those are in the wall of the arteriole. So they're smooth muscle cell, but they are modified, they're specialized, the smooth muscle cell. And they could actually, what do you think the smooth muscles do in the surrounding the blood vessel? They could actually contract or relax, thereby control the afferent arterial diameter, right? In addition to this one first function, that's classic, smooth muscle action, contract, or relaxation. But the specialized part of the granular cells is they also synthesize and release renin. Remember renin we just talked about? That's a hormone, that's an enzyme, basically function as a hormone here, actually going to convert angiotensin 1, right? Angiotensinogen into angiotensin 1. So to generate angiotensin 2, further, generate angiotensin 2, and then going to control the blood pressure. So that's how, we're going to talk more in the context of how the kidney can regulate blood pressure. And that's one of the pathways of regulating blood pressure by kidney. As we promised earlier, right? We said we're going to talk about the blood flow in kidney again, right? The detail, the organized detail level of the artery, what you call them, the nomenclature, how you call them, we are going to go through that in the lab. So I wouldn't spend too much time about it in the lecture, but I just want to point to those two terminologies, right? Those two terms we used for describing the capillaries that surrounding the nephron. We kind of alluded to this earlier, but you can see the blood coming into the glomerulus, right? You have afferent arterial glomerulus, then coming out of the glomerulus as efferent arterial, then feeding those two capillary beds, right? So those are the traditional capillary baths. They're purple in color. They're going to provide oxygen to the surrounding tissues. One set of them associate with the tubules. They're called peritubular capillaries, right? And then, so they're associated with the tubules. The other set are associated with the nephron loop. So they actually can go deep into the medulla, and they run parallel and across, right, perpendicular to the nephron loop, and they have a very special name called the vasa recta. Vasa recta, basically the blood vessels running straight down, right? Vasa recta. So be familiar with those terminology. We're going to mention them again when we talk about how the function of how does the tubules and the nephron loop modify filtrate and in generating urine. Okay, so that's the blood flow, right? And then in terms of the fluid flow, you have initially the filtrate, right, was generated, just directly filtered out of blood plasma. It results in they are in the capsular space. Then they get into the tubules, different segments, different portion of the tubule. They are called tubular fluid, right? So you have different different segment of the tubules. They are tubular fluids flow through those tubules in order, and they are going to then end up in the collecting tubules, number 6, and then drain into the bigger ducts. They're called collecting ducts, and then so they're going to empty it into. Once they are emptied out of the collecting duct, they become urine. Because in this orange step is where all those modification of the filtrate happens. Reabsorption and secretion happens in this part. Once they are out of the capillary duct, so then they are urine. There's no further modification of it. So you have the capillary duct right here. It's just very small. The capillary duct is right here, right? And then you have the number 9 will be the minor calyx and the major calyx, right? It's basically the connecting sinus become bigger and bigger, and then it's called renal pelvis before it drains out into the ureter, and then the ureter goes to the urinary bladder, then urethral, then comes out. So that's the entire flow, where you're going to identify all those structures in the lab. You should be able to be really familiar with all the terminologies mentioned in this diagram and to identify them on the model or diagram. So here's another way to show what I just talked about, right? You have the tubules, then you have the blood vessels, right? Running right next to all the tubules, the blood capillaries, right? And so it's basically the process, the three parts of three process involved in urine formation. The first one is glomerular filtration, right, where the filtrate forms in the capsule, and then as the filtrate flows through the tubules, uh, the modification involved in this process is basically tubular reabsorption and tubular secretion, and that's both of them involves the reabsorption involves the substances, whether it's ions, whether it's a nutrient, right? It's going to be coming back to the blood, right? From the nutrient, from the fluid, from the tubule. And for the tubular secretion to happen, is basically you have active movement of the substance from the blood into the tubular fluid. So this, the reabsorption and secretion in the tubules happened in all parts of the tubule, of the tubular segment of the nephron. It also happens in the along the collecting duct. So that's why you have blood vessels, blood capillaries running right next to all the tubules, is because you are having constant exchange of substances between the tubules and the blood vasculature. So now let's look at the first part of the process, filtration, right? We already talked about the filtration happens basically means the plasma get filtered from the blood from the glomerulus into the capsular space. Okay, so we are going to look at the filtration membrane here, right? So on one side of it is the capillary, on the other side is the capsule space. Okay, the filtration membrane refers to the endothelial cells line up the blood vessel here, the capillary vessel, and that's as I kind of mentioned already, that's the fenestrated type of capillary. You can see the capillaries, there's a lot of windows or holes in the capillary membrane, so to facilitate, for the substances in the plasma to get out, right? So it can filter a lot of the large amount of blood at the same time. But it's the fenestrated capsule. Those holes are still small, so the large protein is going to have to stay inside of the blood. They're not going to be filtered out. The other part of the filtration membrane is formed by the basement membrane, but then also this part here are the filtration slits of the visceral layer. Remember the visceral layer, your interior surface of your baseball glove, right? Covers the capillary, and they have a lot of slits. So they looks like this. They have a slit basically allowing the substances to come out, right? So those slits are formed by basically you have the podocytes, which are the cells covering the forming the visceral layers, right? They are called podocytes. But then they have a lot of filtration slits between the two podocytes, and those those finger-like structure called pedicels, right? They form, so they essentially create a lot of the slits and to facilitate filtrate to form, right? So what's in the filtrate? It includes large amount of water, glucose, amino acids, ions, urea, some hormones, vitamins, ketones, right? Those are all metabolic products, and very small amount of protein, and tends to be small proteins if there are any, because the bigger proteins they cannot get out of the filtration membrane. There's one more cell I want to mention here. It's called the mesangial cell. These are specialized cells in the glomerulus themselves. So the glomerulus, remember the glomerulus are the capillary beds, right, of the arteries. So they localize inside of the glomerulus. What they do is they can uh, phagocytide, basically eat up the macromolecules that's stuck within the basement membrane that kind of stuck in here, right? So if they're stuck here, then they could be eaten up, right, by those mesangial cells. So then they don't clot the filtration membrane. In addition, they can also contract to decrease the filtration area. So those are essentially your filtration area, right? If the mesangial cells decrease the contracts and decrease the filtration area, that's going to decrease filtration. So those are the two functions of the mesangial cells that inside in the glomerulus. Then what determines the filtration? You're probably tired of saying the glomerulus again and again, but this is the most complicated, and I know you guys tend to struggle when we look at the equation, right? Look at the numbers. But bear with me because we have talked about this in the capillary, in the cardiovascular system, talking about what happens in the capillary beds, with the filtration, bulk filtration or bulk absorption, right? Here is similar concepts. So because this is filtration, we're mainly going to talk about the filtration here in the glomerulus. Okay, still the same kind of equation, right? The net filtration pressure is going to decide how much, right, the filtration is going to form, right? So then basically the net filtration pressure, you are going to calculate the hydrostatic pressure. You're going to calculate the pressure that's pushing filtrate out of the blood vessel, but you're also going to calculate the minus. So that will be the hydrostatic pressure from the glomerulus, right? So that's the blood hydrostatic pressure. Then subtract from that, right? Then the blood colloid osmotic pressure. Remember that? Blood colloid osmotic pressure is exerted because of the protein concentration in the blood. We talk about the proteins tend to stay in the blood, not going to be filtered out. So they're going to keep some of the water in, fluid in. So that's the osmotic pressure exerted by the proteins in the blood. So again, I don't know why they are not calling it COP this time. They're calling it OP, but it's the same concept. It's a colloid osmotic pressure in the blood. So that's the pressure keeping the fluid in. Then there's additional like the pressure keeping the fluid in is because there is going to be fluid in the capsule, right? In the capsule, they're in the capsular space, so therefore they exert a pressure pushing the fluid, right, into the capillary, into the glomerulus. So they are called HP, right? Hydrostatic pressure from the fluid in the capsular space. And so they're indicated, they're demonstrated by HPC. C is for capsular hydrostatic pressure. Okay, so they are in the opposite direction of HP glomerulus. So one of those HP is because of the blood pressure, the other one is because of the filtrate pressure. Which one do you think have the higher pressure? Is it the HPG or is it HPC? Of course, HPG is a higher one. If the HPG is not much higher than HPC, then you're not going to be able to form any filtrate because they're opposing each other in opposite direction, right? In order for the filtration to form, you need to have not only HP higher, bigger than HPC, but HPG has to be bigger than OPG and HPC together because those two opposing HPG, right? So you have to subtract HPG, subtract those two from HPG. So exactly this is what happened. How do you calculate NFP? Is from the HPG you subtract those two opposing pressure together, right? So then you end up with the net filtration pressure. And usually you should be familiar with the range, right? So usually the blood pressure here is at the 60, around 60 millimeter Hg, pushing the fluid out. And those two keeping the fluid in, and together they are smaller than the HBG, so you end up with a positive NFP. Okay, so if you notice the blood HP here is even higher than the blood pressure of other systemic capillaries. Why? Right? This is because with this size difference between the afferent arteriole and the efferent arteriole, right? We talk about this being big and this being small. Your artificially basically increase the HP hydrostatic pressure of the glomerulus. So then you end up with a higher, hydrostatic pressure in the glomerulus than of the other capillaries. Okay, you need to really remember this, the three pressure here. How do you calculate its NFP? So you can understand then down the road how then you can regulate the NFP. That therefore regulates how much filtration happens or how much urine you can generate. Well, let's look at how much filtrate we can generate a day. That's a huge amount. That's 180 liters a day. Our kidney can generate that much filtrate. Of course, we don't pee that much, right? That's just showing you how efficient our kidneys are at filter the blood. And so what gets freely filtered, what did not get filtered at all, and what can be limited filtered is all listed here, and we already alluded to. But now let's look at some what influence NFP, right? Based on what we already have, the equation in the last slide. It is driven, a big factor of influencing how you can influence NFP, net filtration, the pressure is by influencing the HPG, that's the glomerular, the hydrostatic pressure in the glomerulus. So the higher the HPG, the higher the NFP. And the NFP also directly going to influence then how much, right, filtrate you can generate per minute. So that's called glomerular filtrate rate. So the higher the NFP, the more filtrate you can generate. That's really obvious. There's a direct correlation between NFP and GFR. But the other one, the indirect correlation of the filtration reabsorption is not that obvious. But you can think about it as, well, the higher the rate, right, of the filtration rate, that's how much you are generating the filtrate. Then basically that means the filtrate has to flow through the tubules, the other part, the tubular compartment, faster. And the faster the filtrate flows through the tubular compartment, the less time they spend in the tubules, and to get to have the filtrate reabsorbed. Right, the shorter the time they spend in the tubules, the less reabsorption. Therefore, you get from you can do to those filtrates. So that's the indirect why you have an increased net filtration pressure results in lower filtrate reabsorption. So that means then if the less you can reabsorb, then the more urine you can generate. You are going to increase the urine output. So next couple slides, we are going to summarize what regulates GFR. Right? So GFR is regulated by the size of the afferent arteriole. Remember, the bigger the afferent arteriole is, the more HPG they can generate, right? Because it's increase the blood flow into the glomerulus, therefore increase the hydrostatic pressure in the glomerulus. So that's one way, right? The smaller the afferent arteriole, then the smaller the HPG. The other one is to alter the surface area of the filtration membrane. And if you can remember, we talk about the mesangial cells can alter the surface area of the filtration membrane. So it's basically through those two mechanisms, right? Then you can alter the GFR. But then there are two ways to alter those two variables, right? One is intrinsic control, one is extrinsic control. Well, let's look at the extrinsic control first. The extrinsic control in general, we have actually used those terms in multiple organ systems already, right? That means the control is exerted by nervous system or hormone. It's not by organ in this organ system themselves. So intrinsic is by kidney, right? Extrinsic is by nervous system, by outside of those factors from outside of the urinary system. And so the hormones we are going to the nervous control is through the sympathetic stimulation. Sympathetic stimulation, if you can remember, can increase the heartbeat, heart pressure, but can also decrease the urinary output, right? That's when we learn about the sympathetic stimulation in AMP1. And how do they do that? Well, it's by vasoconstriction of the afferent arteriole. So decrease the makes the size of the arteriole smaller, and also decrease the surface area of the filtration membrane by contraction of the mesangial cells. Both result in the decrease of GFR. Right? So if the GFR decreases, then you have a decrease in the urine production. That's how the sympathetic stimulation results in decrease of urine production. And the hormonal control is through the atrial natriuretic peptide, AMP. We talked about this, sounds familiar, right? In the cardiovascular system, it is one of those hormones that naturally decrease blood pressure. Part of it, how it decrease the blood pressure is by increase urine production, right? Uretic meaning promoting the urine production. So natriuretic peptide is this hormone increase urine production. How does it do that? It's actually by vasodilation. So increase the size of the afferent arteriole and also the relaxation of the mesangial cells, increase the surface area and results in the GFR increase. Right, so then the increase of GFR, increase of urine output, and then increase loss of fluid because you lose a lot of more fluid in the urine, and you could therefore doing what? You have decreased the blood volume, and decreased blood volume going to result in decreased blood pressure. That's how an AMP can lower blood pressure.